Hey guys, today we're going to talk about the scientific method. The scientific method should be something that you're comfortable with already because you should have learned it almost every single year throughout your entire science career. So today we're just going to review what it is and how it works in a very quick video. So the definition of the scientific method is a method of procedure consisting in systematic observation, measurement, and experiment, and the formulation, testing, and modification of a hypothesis. So, real people words. It is a process to go through that consists of observing, measuring, and experimenting to try to test or modify a belief. So you have a hypothesis. I think something happens for a certain reason. The scientific method is the way that you go about testing to see if you can support that idea or refute that idea. Its purpose is to help scientists use critical thinking to search for answers. So they don't want to just start an experiment without making a plan. This gives them steps to follow, in addition to ideas that they have to cover and talk about before they can move forward with that plan. The scientific method is an ongoing process. We have this huge diagram, and it's a big flow chart all in a circle. So the first thing that you have to do, the very first idea is up here. This is number 1. Make an observation. What do I see? What do I notice? And what do I observe? The second thing, think of an interesting question. Why does that pattern or that idea happen? Can I think of a theory or a question that would support or tell me why that item occurs? 3, formulate your hypothesis. What are the general causes? What do you think makes it happen? 4, develop a testable prediction, so make a testable statement. 5, gather data to test your predictions, and then we can either go up into the develop general theories, or we can follow this purple arrow. And if we follow this purple arrow, essentially what we're saying is our data did not support our prediction, so we're going to refine or alter our hypothesis and retest. Refine and alter and retest. Refine and alter and retest. And really, this loop, we can go through hundreds of times for an experiment for 2 reasons. The first reason is to provide an ongoing data set to help us support our theory. And once we have hundreds of examples of data, then we develop our general theory. These theories do not occur out of nowhere. If you were to explain a theory and say, here is my data, I did this experiment twice, scientists may not take you seriously. So instead, what they do is they go through this process of alter, expand, or reject their hypothesis. If you gather data the first time around and it supports your prediction, you then expand on your hypothesis. Develop your testable prediction, test it again, expand, develop, test. Maybe refine, develop, test. Refine, develop, test. The whole idea is you go through this cycle. You want to collect multiple sets of data to support your hypothesis. If you go to a counselor, if you go to a scientific board and want to publish a paper, if you say, I have thousands of data sets that support my idea, you will be taken a lot more seriously than saying, well, I did it once and it worked. So multiple data sets help us create this idea of a theory. So the importance of repeating the process. I've already explained this, but I want to make sure you write this down. The more evidence you have to support your conclusions, the better. That means that having multiple trials and data sets makes it harder for other people to debate your findings. If you say, I've done this 1,000 times and every single time it supported my idea, they have very little room to say, are you sure? But if you only do it once or twice, they can question you and say, well, maybe you don't have enough information yet. But if your data does not support your conclusion, scientists don't just give up. They go back and adjust their original hypothesis based on the information that they found and retest that new idea. So the fun thing about being a scientist is that even if you're wrong, you can still be right in the end because you can adjust your thinking, you can adjust your process so you then support your data and support your idea. So we're going to meet in class, and when we meet in class, we're going to talk about this whole process of the scientific method. We're going to talk about the making of observations, why it's important, and we're going to use that with the following slide. Here are some other important terms that you need to know, and these are ones that you should have heard before, so again, just a review. Independent variable. This is the variable that is controlled by the experimenter. This is the one that you are saying, I'm going to adjust the amount of sunlight that a plant gets in the day. The dependent variable is a variable that is being measured by the experimenter. So when I adjust the amount of sunlight the plant receives in one day, how does that affect the height that the plant grows? Control is the basic trial that you use as a reference point. If I just let the plant receive the normal amount of sun, I put it outside and it gets however many hours of daylight and nighttime that the normal day provides, that may be the control. The constants are variables that you keep the same. So these two are always confused. Control is a basic trial that you use as a reference point. Constants are other variables that you can test, you are just choosing not to right now. Okay, so the whole idea of a plant getting a certain amounts of sunlight. Other constants may be the amount of water that the plant receives, the amount of fertilizer that the plant receives, the type of soil that you use to plant the plant in. Those are things you could eventually test. You just don't want to test them at the same time as the amount of sunlight. And then the last one, we're going to go back up here to repeated trials. Essentially, you're just collecting data more than once. Now we're going to pretend that we do repeated trials, and what we're going to do instead is possibly share data throughout the class. Because if we have 8 groups doing the same experiment, technically that's the same thing as doing the experiment 8 times with one group. So instead of repeating the trials ourselves due to time constraints, we are actually going to share data within the class. Okay, so these are the other important terms that I want you to know. We are going to be identifying these in class tomorrow also. If I give you an experiment, I want you to be able to tell me which the independent variable is, the dependent variable, and etc. Okay, so I will see you tomorrow when we can talk about this a little bit more in depth, and we can practice identifying the parts of the experiment and the scientific method. See you tomorrow.